Hey everyone, Dr. D here, and in this video we are going to be doing an exam 2 review for Biology 2420. That's microbiology for non-science majors at TCC. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's get started. This exam 2 covers chapters 6, 7, 9, and 11. So those are the handouts that you want to study. So let's go ahead and start with chapter 6, microbial genetics. Okay, obviously we want to know all these main terms here like genome, which is the sum total of genetic material of an organism. Genomics being the study of an organism's entire genome. Remember that you have chromosomal DNA as well as non-chromosomal DNA, or also known as extra-chromosomal DNA. This includes in prokaryotes plasmids, which are tiny extra pieces of circularized DNA. In eukaryotes, you have extra-chromosomal DNA in the mitochondria and in the chloroplasts as well. The genomes of cells are made up of DNA, but remember that viruses can have a wide variety of different types of genomes. They could be DNA genomes or RNA genomes. Okay, where is the genome? In a eukaryote, remember the chromosomal DNA is in the nucleus, while the non-chromosomal DNA is found in the chloroplasts and the mitochondria. In bacterium, there's usually one circularized chromosome, and that's its chromosomal DNA. And then outside of that, you usually have plasmids, small circularized DNAs. And then in viruses, inside of the capsid protein coat, you have either a DNA genome or an RNA genome. Learn all of these terms. Remember what a chromosome is: a distinct cellular structure composed of neatly packaged DNA, usually double-stranded DNA. Eukaryotic chromosomes are found in the nucleus. Bacterial chromosomes, there's usually a single circularized chromosome floating around in the cytoplasm. So remember what a gene is. You should know that a gene is a basic informational packet. Usually a gene is the necessary information to produce a particular protein. Remember that? Genes, structural genes code for proteins. So that's important to remember. Remember what genotype refers to. Genotype refers to the sum of all types of genes. So if the genotype could be, you got a brown eye allele from one parent, you have a brown eye allele from another parent. Well, those genes that make up your eye color are your genotype, while your phenotype is how those genes express themselves, right? So if you have brown eyes, that's your phenotype. But if you have the genes for brown eyes, that's your genotype. Remember that? And then we talked about variation in genome size. You should know that prokaryotes have fewer genes and less DNA in general than eukaryotes. For example, E. coli has about 4,000 different genes, while a human cell has about 23 to 25, by some estimates, 1,000 genes. E. coli only has a single chromosome, while humans have 46 chromosomes. Also, remember, prokaryotes have circularized chromosomes, usually, while eukaryotes have linear chromosomes. Remember, the basic unit of DNA is a nucleotide. You should know the parts of a nucleotide include a phosphate group, deoxyribose sugar, a nitrogenous base. Remember the pairing that occurs in DNA. A's or adenine pairs with T's, thymine. G's or guanine pairs with C's, cytosine. Okay. Remember that DNA strands are anti-parallel. Okay, you should know the central dogma of genetics. This refers to information flowing from DNA to RNA and then from RNA to protein. The process of copying a gene from DNA to RNA is called transcription, while the process of reading the RNA message to produce a protein is called translation. And as I mentioned before, there's a gene to protein connection, right? Genes code for proteins. DNA is mainly a blueprint that tells the cell which kinds of proteins to make and how to make them. The double-stranded DNA codes for proteins, and the way it does so is by these 3-letter codes called triplets or codons, triplet codons. These 3-letter codes are copied to mRNA, and then these 3-letter codons are translated to protein. Copying double-stranded DNA to mRNA is called transcription. Translating that transcript to protein is called translation. Here you can see the genetic codon table. This is every possible RNA codon in the cell. You have 64 total codons, and these codons code for the 20 amino acids. For example, UUU codes for the amino acid phenylalanine, but so does UUC. Okay, you have 64 codons, only 20 amino acids that they code for. This is why you have multiple codons coding for the same amino acid. One of the codons, AUG, is the start codon, so we should know about the start codon. And 3 of the codons, UAA, UAG, and UGA, are the stop codons. So we should know that AUG is the start codon. We should know that these 3 are the stop codons. Okay, and we should know what codons are. Codons are triplet codes like this that code for particular amino acids. Okay, next, we discussed operons. Remember, you should know that an operon is a coordinated set of genes regulated as a single unit. Remember inducible operons? You should know that inducible operons are usually off, but can be turned on when you add an inducer. This contains genes that make proteins that help metabolize nutrients. So inducible operons are usually operons for making proteins that metabolize or break down nutrients, such as the lac operon is a inducible operon because when it's induced, it helps to break down or metabolize lactose. Repressible operon, on the other hand, these contain genes coding for enzymes that synthesize amino acids and synthesize things in the cell. Usually, these operons are on, these genes are on. However, they can be repressed when there's plenty of that particular amino acid present. All right. Remember, we wanted to know how the lactose or lac operon works in bacterium. You have a repressor protein which normally represses the lac operon, preventing the enzyme that can copy the operon and produce mRNA, RNA polymerase, from doing its job, right? It gets in the way because it binds to the operator here. When lactose is present, lactose can serve as the inducer by binding to the repressor and causing inactivation of the repressor. Once the inactive repressor lets go of the operator, the RNA polymerase can bind to the promoter and promote transcription, right? Transcription meaning the production of mRNA, which is a copy of these structural genes. Remember, you have functionally related genes, all kind of concatenarized here. They're end to end. You got structural gene 1, structural gene 2, structural gene 3. Remember what these genes are? One of them is beta-galactosidase. Remember the special enzyme that breaks down lactose? Okay, and then you produce the 3 different proteins, right? You produce the 3 different enzymes. Okay, and then once lactose is not present anymore, that causes the the repressor to become activated. The repressor binds to the operator, turning off the genes. Okay, so again, the lac operon is an example of an inducible operon, and this is its mode of action. Again, remember the differences between inducible and repressible operon. Repressible operon are usually in the on position, but they can be turned off. An inducible operon is usually off, but can be turned on. Okay, so let me show you an example of a repressible operon, which will only be turned off when the nutrient is no longer required to build. Okay, if excess nutrient is present, it serves as a co-repressor to block the action of the operon. Let me show you how it works again. Here you have a repressible operon. This is the one for producing the amino acid arginine. Here, the RNA polymerase binds to the operator, or the promoter, I should say, and then it copies the genes. It copies structural gene 1, structural gene 2, structural gene 3. It copies these genes into mRNA. That mRNA is translated into these enzymes that synthesize arginine. That's great. However, if excess arginine is present, if a lot of arginine accumulates, then the arginine can actually bind to the inactive repressor, activating the repressor, serving as what's known as a co-repressor, which binds to the operator, shutting off gene transcription. Okay, so here the gene is normally on, the operon is normally on, but accumulation of arginine causes it to serve as a co-repressor, turning on the repressor to shut down the operon. Okay, and this makes sense because normally arginine is hard to come by, so the cell wants to create arginine by producing these three enzymes. However, when arginine accumulates, that arginine can shut off the transcription, of these genes that produce these enzymes because no longer needed. You have excess arginine. All right, next we talked about recombination. Recombination is an event in which one bacterium donates DNA to another bacterium, and the end result being a strain that's different from both the donor and the recipient strain. The recombination can include a plasmid that's exchanged, which, remember, plasmids are extra chromosomal DNA usually in prokaryotes. And the recombinant is defined as any organism that contains and expresses genes that originated in another organism. There's different mechanisms by which these prokaryotes do this, and they're called horizontal gene transfer, which is any transfer of DNA that results in organisms acquiring new genes that did not come directly from the parent organisms. Remember, plasmids are small circular pieces of DNA that are non-chromosomal or extra chromosomal. They contain a few dozen genes, okay, at most a few dozen genes. They're not necessary for survival, but often carry useful traits such as genes for antibiotic resistance or for producing toxins and such. Usually they're found in bacteria, but sometimes in other organisms, such as fungi as well. Okay. Now, these were the three modes of horizontal gene transfer that we talked about in class. There's conjugation, transformation, and transduction. So what I want you to know is that conjugation is done through the sex pilus. Remember, when an F plus cell produces the sex pilus and sends DNA to an adjacent cell, that cell does not need to be of the same species. Transformation, in which free DNA, like an actual double-strand DNA, gets taken up by a competent recipient cell. And transduction, where a bacteriophage, a virus, actually helps with the horizontal gene transfer by incorrectly packaging donor DNA. Again, in conjugation, you should know this is a mode of genetic exchange in which a plasmid or other genetic material is transferred from a donor to a recipient via a direct connection. Remember the sex pilus. One of the best understood conjugated plasmids is F factor. Remember that if a cell has the F factor, that means that it can make the sex pilus. So if the cell is F positive, that means it has the F factor. In this case, the F factor is on this plasmid. That allows this cell to make the pilus. And then that after the pilus is formed, there's a process by which this plasmid can share its information with the other cell. And now both cells are considered F+, which means now the recipient cell or the recombinant cell is able to make its own sex pilus and share its own genes. And remember, this is different from a HFR transfer or high frequency of recombination transfer, where in this case the F factor is in the genome, the circularized genome of the host cell, but the same process is triggered where a sex pilus is formed, but this time the chromosome of the host is shared, right? And usually not the whole chromosome, but fragments of the chromosome can be shared with an adjacent cell, and then there's this recombination that occurs, and now you have this like hybrid organism here with some of the genes of the donor. What are resistance plasmids? Resistance plasmids carry genes for antibiotic resistance. Transformation, how does this work? This is when naked double-stranded DNA is actually taken up by cells called competent cells. Competent cells are cells that are capable of taking up naked DNA from the environment. That naked DNA can recombine into the host's genome, forming a recombinant. Okay, this is called transformation. But remember, transformation requires competent cells, cells that are capable of accepting genetic material through transformation. Lastly, there was transduction. Remember here, this process requires a bacteriophage to serve as the carrier. The bacteriophage usually infects a cell. Here you can see the bacteriophage infects a cell with its DNA. The bacteriophage produces its own components. The bacteriophage should produce more bacteriophage, right? So assembly occurs. However, this virus on the right, you can see that it actually packages some of the host's DNA instead of viral DNA. And then when that virus injects its DNA into a new host, remember this is just DNA from the donor chromosome. This is not real viral DNA. And if there's a recombination that occurs, now you have incorporation into the genome of the chromosome of this bacterium, and you have a recombinant cell formed. So this is known as transduction. Okay, we talked about transposable elements. These are called jumping genes, where genes can shift from one part of a genome to another. We talked about mutation. Mutation is any change to the nucleotide sequence in the genome. It's a driving force of evolution. When we're talking about mutations, the normal gene is called the wild type gene, while the gene with the change is called the mutant gene or the mutant strain. There are spontaneous mutations, which are random changes in the DNA just arising from errors in DNA replication. They occur randomly and not by any particular reason. And then induced mutations, which result from exposure to physical or chemical agents that damage the DNA, known as mutagens. The Ames test is a test that is done to look for mutagens. What we have are a culture of bacterium that cannot grow in the presence of histidine, and when you plate them, the very few bacterium grow, and these bacterium that do grow are bacterium that have undergone what's known as back mutation. They've actually randomly and spontaneously fixed the histidine mutation. Now, if you treat these bacterium with a mutagen, that's going to increase the rate of back mutations, and you're going to have a lot more colonies growing on these plates. Okay, so the plates on the right have the test chemical, the potential mutagen, and the plates on the left have no potential mutagen. So if there's a difference in number of colonies in the second plate here, then that shows that this is a mutagen that you're dealing with. If both of these plates have very few and comparable numbers of colonies, then it's not a mutagen. So the Ames test is a very interesting test which can screen for mutagens. Remember too that photoactivation is light repair of damage caused by ultraviolet radiation. Remember that ultraviolet radiation causes adjacent thymines or adjacent cytosines to fuse, forming thymine-thymine dimers or cytosine-cytosine dimers. And light enzymes, or enzymes that respond to light, can actually repair those dimers. This is an enzyme called DNA photolyase, which can detect and attach to the damaged area and fix those dimers. Cells also have what is known as mismatch repair, a repair system that can locate mismatched bases, so mutations essentially, that were missed during proofreading, and then fix them. They can fix them. All right, that leads us to the end of chapter 6. Let's take a short break time with Gizmo and Wicket and get right back with it with chapter 7. All right, let's do it. All right, welcome back from break time with Gizmo and Wicket. Let's carry on with viruses and prions, chapter 7. Remember that all types of life are infected with viruses. No one is spared, not even the archaea are spared from infection with viruses. Remember that viruses are not alive. Remember, because viruses are not alive, they are either described as active or inactive. One of the main reasons they're not alive is because they are unable to multiply independently from a host cell. Okay, remember again, viruses are not cells. They are not alive. They are obligate intracellular parasites of all different kinds of life, of different types of cells. Okay, they are inactive outside of the host cell and active only within host cells. The basic structure of a virus is a protein shell called a capsid, which surrounds a nucleic acid core. Remember, they are ultra microscopic in size, so we cannot resolve them with our light microscopes in the lab. They can have genomes that are either DNA or RNA, but not both. They can have double-stranded DNA, single-stranded DNA, single-stranded RNA, or even double-stranded RNA genomes. They carry molecules on their surface. Remember, these are often called S proteins that determine specificity for attachment to host cells. They multiply by taking control of a host cell's genetic material and regulating synthesis and assembly of new viruses, also hijacking the ribosomes. They usually lack enzymes for most metabolic processes. Remember that at the core of every virus is what's known as the nucleocapsid. This includes the nucleic acid as well as the protein capsid coat, and that some viruses are what is called naked, which means that it's just a nucleocapsid, while other viruses have a covering known as an envelope. So these are known as enveloped viruses. Okay, remember the viral components? Viruses have capsids, which are a protein shell surrounding the nucleic acid. And this comprises a nucleocapsid, which includes the capsid as well as its genome, the nucleic acid together. Every virus has a nucleocapsid core. Some viruses have an envelope, which is usually a modified piece of the host cell's membrane. Remember I told you all where the virus gets its envelope from? It steals it from the host. Then viruses also have spike proteins on their surface, be it naked viruses or envelope viruses. These spike proteins allow viruses to dock specifically to receptors on the host cells. So here you can see the spike proteins on top of a naked nucleocapsid. Here you can see the spike proteins on the envelope, on the surface of a enveloped virus. And the term virion refers to a fully formed virus. Capsomeres are the individual proteins that make up a capsid. So these are identical protein subunits that spontaneously self-assemble to form the capsid. Now, capsids can have different arrangements, right? There are helical capsids, which are rod-shaped, which are made up of a continuous helix of capsomeres. And then there are icosahedral capsids, which are three-dimensional, 20-sided, almost like a 20-sided dice. And these are the two most common capsid shapes. Here you can see a helical capsid of the tobacco mosaic virus. See how the nucleic acid is protected on the inside, and those capsomeres, those individual capsomeres, wrap around in a helical fashion to form a rod. Here you can see a icosahedral capsid, which forms a 20-sided die version, a 20-sided dice structure. Now, you can have naked helical viruses, enveloped helical viruses. You can have naked icosahedral viruses, enveloped icosahedral viruses. Bacteria can be infected with viruses as well. And the viruses that infect bacteria are known as bacteriophage, which look like this. Okay, this is known as a T-even phage. In this case, it's the T4 bacteriophage. It actually has it's really neat because it's a complex capsid. Complex capsids, only found in the viruses that infect bacteria, may have multiple types of capsid. So, for example, they can have an icosahedral head with a helical tail. See, so that's known as a complex capsid. Okay, again, you can have naked viruses. You can have enveloped viruses. The envelopes can be loose. You see, like this virus here. The envelope can be loosely associated with the virus. Some viruses have envelopes which are tightly associated with the virus. Again, the viral envelope is composed of membrane from the host because the virus kind of stole the host's membrane. And then there are spike proteins protruding from the membrane. And these spike proteins allow for attachment to the host cell. Remember, viruses have a genome. This genome can be either DNA or RNA, but not both. The number of viral genes is often but not always small compared that of a cell. For example, hepatitis B virus only has 4 genes, and Pandora viruses have over 2,500. So the number of genes varies quite a bit, but it's usually fewer genes than a cell, a bacterial cell. Now, the genomes of the virus can either be DNA or RNA. And if it's a single-strand RNA, there are 2 types of single-strand RNA, either positive sense single-strand RNA or negative sense single-strand RNA. What you need to know is that positive sense single-strand RNA can be immediately translated to protein because this is the same type of RNA as mRNA. It's the same sense as mRNA, whereas negative sense mRNA or negative sense RNA genomes cannot be immediately translated to protein because they are not the same sense as mRNA, and they need to be kind of copied into positive sense RNA before they can be translated. Okay, and remember that the Baltimore system for viral classification categorizes the different types of viruses based on their genome. So class 1 viruses have double-strand DNA, class 2 have single-strand DNA, class 3 have double-strand RNA, class 4 have that, remember, the positive sense single-strand RNA, which can be readily converted into protein. Remember, positive single-strand RNA is ready to convert into protein. Whereas class 5, this makes up your negative sense single-strand RNA, which cannot readily be made into protein. It needs to be converted to positive sense before it can be used to make protein. Then class 6 are known as the single-strand RNA reverse transcription viruses, also known as the retroviruses. And then lastly, there's the double-strand DNA viruses that also need reverse transcriptase. These are also known as the gapped DNA viruses. So there are 7 classes of viruses based on their genomes. Now, there are other components of the virus particle as well. For example, specific enzymes for the virus. The virus may code for polymerase enzymes. Polymerases are special enzymes that synthesize either DNA or RNA. Replicases, which copy RNA. And the special reverse transcriptase enzyme, which can actually synthesize DNA from RNA. Remember, these are found in class 6 and 7 viruses. All right, next moving on to the multiplication cycles in animal viruses. This is known as the life cycle of an animal virus, but remember, this term life is used loosely here because this is, the viruses are not alive, but this is still known as a life cycle of a virus. Remember the steps here: adsorption, penetration, uncoding, synthesis, assembly, and release. So let's go through each. Adsorption, remember, not absorption. Adsorption refers to attachment. The virus needs to attach to specific receptors on the host cell, and it does so with its specific spike proteins on the surface. And this dictates its host range. So, for example, hepatitis B only infects liver cells because only those liver cells have the specific receptor on their surface that's recognized by the hepatitis spike proteins. So this means that hepatitis B has a very restricted host range. It only infects a particular tissue on a particular species of animal. Whereas other viruses are moderately restrictive, such as polio, which infects intestinal and nerve cells of primates. And then there are broad host range viruses like rabies virus, which can infect all mammals. Okay, next, penetration and uncoding. A lot of times, the penetration occurs by endocytosis. This means that the virus, makes its way in by the cell membrane engulfing. Okay. And then there's uncoding. Uncoding refers to when the DNA and the enzymes inside of the capsid coat are, released. They're uncoded. They're released into the cell. Next, during the synthesis stage, the genome is replicated and the proteins are produced for the virus. You should know that RNA viruses replicate in the cytoplasm, while DNA viruses replicate in the nucleus. All right, so again, there's adsorption, penetration, uncoating, synthesis, and then assembly. Assembly involves the virus forming new virion by putting the DNA or RNA nucleic acid inside of a capsid coat, forming the nucleocapsid, and then also making sure to package all of the other enzymes and forming new virion, right? This is assembly. And then we're ready for release. During release, enveloped viruses are liberated by budding or exocytosis. Remember, this is when the virus pushes its way through the cell membrane, taking a part of the cell membrane with it as an envelope. While naked viruses, they actually lyse the cell upon release. So naked viruses lyse the cell upon release, while enveloped viruses release by budding. That's how they obtain the membrane of the host cell, the envelope. And remember that this can lead to damage to the host cell, known as cytopathic effects, or CPEs. Okay, recall that viral infections can also become persistent. This is when the cell harbors the virus, but the virus, does not immediately lyse the cell or exit the cell. Viruses can form what's known as a provirus, where the viral DNA incorporates itself into the DNA of the host chromosomes. You can form a chronic latent state, where after a short period of inactivity, the virus gets reactivated and reemerges. Think shingles after a chickenpox infection. You should know that viruses can also cause cancer. Up to 13% of human cancers are caused by viruses. These are known as oncogenic viruses. And a cell that has become cancerous is known as a transformed cell. The virus carries genes that directly cause cancer, or the virus produces proteins that induce a loss of growth regulation in the cell. These are the two ways by which viruses can cause cancer in a cell. Then remember that some viruses infect specifically bacteria. These are known as bacteriophage. Remember that lunar lander-looking virus? Usually these are double-stranded viruses. Okay, these are known as the T-even bacteriophages. So for example, T followed by an even number, such as the T2 phage or the T4 phage. The most studied is probably the T4 phage. And remember, it has a complex capsid with an icosahedral head and a helical tail fibers and tail pins. And these viruses are interesting because the capsid remains outside of the bacteria, while the DNA is injected inside of the bacterial cytoplasm. And when the bacteria is infected with the virus, remember, the virus can sometimes have two different life cycles. One's known as the lytic cycle, in which the virus adsorbs, penetrates, replicates, assembles, matures, and lyses the cell. Or the other option is something known as the lysogenic state. Remember, where the virus penetrates the bacteria with its DNA. That DNA incorporates itself into the host DNA, forming a prophage, and then this bacteria is able to just grow, and it's kind of like a latent infection where it's a dormant state. Hi, Wicket. Hey guys, Wicket has come to say hi. Hi, Wicket. Hi, Wicket. Well, gosh. Hey, you guys, Wicket's come to say hi. Here's sweet Wicket. He's pawing at the screen, making sure that, everything checks out. He's quality control around here. We need to make sure that everything is good. He's checking my work. Thanks for coming to say hi. Sweet Wicket. Again, bacteriophage life cycles can either be lytic or lysogenic. During the lytic phase, the bacteriophage ends up destroying the cell, lysing the cell. And during the lysogenic stage or lysogenic cycle, bacteriophage becomes incorporated into the host cell DNA as a prophage. And this allows the bacteria to divide. And as long as things are going well, remember, as long as things are going well, the virus will lay low and allow the bacteria to live its life. But once the bacteria is starving or has low nutrients or other insults, that's what activates the cell to, oh my goodness, Wicked's come to say hi again, you guys. This is what triggers the prophage to release itself from the host genome and to release itself and enter a lytic cycle to explode the cell. Oh my goodness, he's scratching the monitor now. He really wants attention and pets. I know, buddy. Be careful. Careful, Wicket. So these lysogenic phage, these lysogenic viruses are known as temperate phages. They have the ability to undergo adsorption and penetration, but do not immediately undergo replication or release. This is because they can enter the lysogenic life cycle. Remember, where the prophage forms, the viral DNA integrates into the bacterial host genome and allows the host to replicate. And every time the host replicates and copies itself, it also copies the viral genome, the prophage. This is to say until there's an induction. This means that the prophage becomes activated, and this will lead to viral replication and lytic cycle. This happens when the bacteria is, short on food or resources, or it gets UV damage or something else. There's an insult to the cell, and this can lead to induction. So you can think of it as leaving lysogeny, leaving the lysogenic cycle and entering the lytic cycle. Okay, as you all know, viruses cause human health problems. They cause diseases. Some do not result in hospitalization, such as colds, chickenpox, the flu, herpes, and warts. Some have very high mortality rates, rabies and Ebola, and others can lead to long-term debility, polio and neonatal rubella. All right, now moving on to prion diseases. Prions, remember, are another non-cellular infectious agent. They are made up of only proteins, and they cause spongiform encephalopathy, which means severe brain damage that results in a sponge-like brain, right? So cavities to form in the brain. Creutzfeldt-Jakob disease is an example of a prion disease in humans. It infects the central nervous system, causing gradual degradation and death of the brain. It is transmissible by unknown mechanism, though usually it can be traced to ingestion of brain material. Several animals are victim of similar diseases. In sheep, mink, and elk, you can see scrapie, and in cows, you can see mad cow syndrome or bovine spongiform encephalopathy. The exact mode of infection is unknown for prion disorders. However, you should know that it's, proposed that there is a misfolded prion protein, and this causes a cascade of misfolding where all the prion proteins, once one or two prion proteins are misfolded, this causes a cascade where the rest of the brain normal prion proteins are also misfolded. Lastly, we talked about viroids, which are virus-like agents that parasitize plants. However, they are not viruses. They are composed only of naked strands of RNA. They do not have a capsid or any other type of coating, and they can cause problems in plants. And that's it for Chapter 7. Let's go ahead and take another break time with Gizmo and Wicket and head over to Chapter 9. All right, welcome back from break time with Gizmo and Wicket. Let's start Chapter 9, Microbial Nutrition and Growth. So what do we need to know here? We need to know about nutrition. We need to know that these are the most common elements in living cells: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So these are required in large quantities. These are macronutrients. We need to know that micronutrients include the trace elements, which are needed in much smaller amounts. Obviously, we talked about inorganic nutrients and organic nutrients. Know the difference between those two. So it's important that you know that 70% of the cell content is water. So most of your weight is water weight. However, 97% of the dry weight after water has been removed is composed of organic compounds, and 96% of that weight is composed by these 6 elements alone: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So these are the big 6, right? These are the 6 most important elements that make up the majority of your body weight. You should know that heterotrophs are organisms that must obtain their carbon in organic form by digesting organic material. Whereas autotrophs are self-feeders. They obtain their carbon as CO2. So they do not need to ingest organic matter. They get their carbon from the air. Okay, we definitely need to know the difference between phototrophs and chemotrophs. Phototrophs are microbes that photosynthesize. They get energy from sunlight. Whereas chemotrophs are microbes that gain energy from chemical compounds, from eating chemical organic molecules. You should know what a photoautotroph is, right? Photo meaning energy from the sun, and autotroph meaning self-feeder, so CO2 from the air. You should know what a chemoautotroph is. The chemo means energy from organic material, while the auto means carbon from carbon dioxide. What's a chemoheterotroph? Energy from organic materials and carbon from organic materials. So the first part of this word refers to where you get your energy. The second part of this word refers to where you get your carbon. You should know that saprobic microorganisms, or saprobes, are decomposers, such as fungi and some bacteria. You should know that saprobes, they digest organic material on the outside of the cell, and then they transport the nutrients into the cell. You should know that there are obligate saprobes and facultative saprobes. You should know there are facultative parasites, which can be saprobes that can infect hosts sometimes. We should know about parasitic microorganisms. Parasites live in or on the body. That's where they get their nutrition from. They become nutritionally dependent on the host. Obligate parasites are unable to grow outside the host. Obligate intracellular parasites are unable to live outside of the host cell. Do you recall diffusion? Substances moving from an area of high concentration to an area of low concentration. This is a spontaneous process. Do you guys remember osmosis is the diffusion of water across a selectively permeable membrane? You should know how osmosis works. So water moves from where water is more pure to where water is less pure. So for example, into the bag. Remember tonicity? Isotonic conditions, hypotonic conditions, hypertonic conditions. We need to remember this. Okay, so this is an important thing for the exam. In an isotonic solution, the solute concentration outside of the cell matches the solute concentration inside of the cell, so there's no net flow of water. In a hypotonic solution, there's less solute outside of the cell than inside of the cell. This results in water entering the cell, and if it's a cell without a cell wall, this causes the cell to lyse or break open. But if it is a cell with a cell wall, this causes the cell to become firm. Okay. Conversely, in a hypertonic solution, this means the solute concentration outside of the cell wall is higher than the solute concentration inside. That results in water rushing out of the cell. There's a net flow of water out of the cell. This causes an animal cell to shrivel, but it causes a different phenomenon in bacterial cells. It's called plasmolysis. This is when the cell membrane shrivels away from the cell wall. Okay, remember what active transport is? This is when substances are moved across the membrane against the concentration gradient, against diffusion. This requires energy. Okay, and lastly, we talked about endocytosis. This is bulk transport into the cell. So endocytosis is bulk transport into the cell by forming vesicles. Phagocytosis is when solids are brought in through vesicles, and penocytosis is when liquids or solutions are brought in via vesicles. All right, moving on to environmental factors that influence microbes. Temperature. You should know that there are cardinal temperatures, which is the range of temperatures for growth of a given microbial species, minimal temperature, maximum temperature, and optimum temperature. And this differs for different types of bacterium. Okay, you should know these classifications, okay? Psychrophiles grow in cold conditions, right? Whereas psychrotolerant bacterium grow in relatively cool conditions. Mesophiles grow best at room temperature to body temperature. Thermophiles grow best higher than body temperature, and then hyperthermal philes grow in boiling solutions, very hot environments like sulfur springs. So if I were to ask you on the exam, which temperature type do you think causes the most human disorders? Which one would you think that is? And if you guessed, mesophiles, that makes sense. Bacteria that grow best at the body temperature are the most likely to cause human diseases. Okay, remember, oxygen is quite toxic to bacterium. Oxygen can form what's called singlet oxygen, which is extremely reactive and toxic to cells. Okay, oxygen can form superoxide ion, hydrogen peroxide, and hydroxyl radicals, which are destructive to cells. And cells use enzymes to scavenge and neutralize these reactive oxygen species. Remember the enzymes we discussed in class? Superoxide dismutase enzyme to neutralize superoxide ion, and catalase or peroxidase enzymes, which neutralize hydrogen peroxide. Only bacterium that have superoxide dismutase and catalase are able to grow in the presence of oxygen. Aerobes are bacterium that grow in the presence of oxygen. Facultative anaerobes are bacterium that do not require oxygen but can grow in the presence of oxygen. Microaerophiles do not like to grow in normal atmospheric concentrations of oxygen. They prefer low concentrations of oxygen. Anaerobes are bacterium that prefer to grow in the absence of oxygen and sometimes can be killed in the presence of oxygen if they do not have those enzymes. Aerotolerant anaerobes, these are bacterium that do not utilize oxygen. They can survive and grow to a limited extent in its presence. Okay, what are capnophiles? Capnophiles grow best in the higher CO2 concentrations. Then there are pH preferences for different bacterial cells. Bacteria that prefer neutral environments are called neutrophiles. Bacteria that prefer acidic conditions are acidophiles. Bacteria that prefer basic conditions are called alkalinophiles. Obligate acidophiles, as their name suggests, require an acidic environment for growth. Alkalinophiles, they grow in basic environments. Bacteria that can live in high osmotic environments, high solute concentration, are called osmophiles. Halophiles prefer high concentrations of salt. Obligate halophiles must grow in high salt. Facultative halophiles grow with or without the presence of high salt. Okay, next, hydrostatic pressure. Barophiles are bacterium that can grow in high actual pressure, atmospheric pressure, so you can find them deep in the ocean. Okay, remember we discussed in class that bacteria also need a certain amount of moisture to grow on. If it's too dry a condition, then there's not enough water on the external surface, and this can cause the cell to not be able to grow. Next, we talked about associations between organisms. There are symbiotic organisms and non-symbiotic organisms. Symbiosis can either be friendly or not friendly. Remember that. Just because something is called symbiotic does not mean it's friendly. Symbiosis means organisms live in close nutritional relationships. So this could be friendly in the case of mutualism, where both members benefit, or it could be not so friendly in the case of parasitism, where the parasite is benefited, but the host is harmed. In commensalism, remember this is kind of a happy medium of the two, the commensal bacterium benefits, or the commensal organism benefits, the other member is not harmed. Think of your gut bacterium, for instance. Non-symbiotic relationships, this is when organisms are free living. Relationships are not required for survival. This can either be synergistic or antagonistic. During synergism, members cooperate and share nutrients. During antagonism, some members are inhibited or destroyed by others. Now, biofilms are the epitome of synergy. In biofilms, you have mixed communities. This means different species of different kinds of bacteria and other microbes all living in the same environment in this secretion, this biofilm goo. Okay, remember how bacterial biofilm forms? Pioneer microbes colonize a surface. Once there are enough microbes or a quorum of microbes, they can release the biofilm material. Other species join and contribute to making this extracellular matrix and or participate in quorum sensing. And then the mature biofilms may stop or minimize quorum sensing. Biofilms serve as a constant source of bacteria that can escape and become planktonic again. This is kind of the formation, the steps to formation of biofilms. And do you remember how bacterium divide? They grow by a process known as binary fission, not mitosis like in eukaryotic cells. You should know the steps of binary fission. A cell grows, it then replicates its circularized chromosome, then a septum forms. A septum forms at the center of the cell, and then that results in two cells. Now, the generation time is known as the doubling time. This is the amount of time it takes for this process of binary fission to occur, so for one cell to become two. And remember, the way this works is in an exponential way. The bacteria divide exponentially, so they're 2 to the power. Okay, again, microbes exhibit exponential growth. And do you guys remember how we talked about growth curve of bacteria in culture, also known as batch growth of bacterial growth? At first, microbes in a batch or culture experience a lag phase in which they do not multiply. They are becoming adjusted to their new environment. And then bacterium switch to a exponential growth phase where they are growing exponentially. And then at some point they exhaust the space and the resources and they enter a stationary phase where no bacterial growth is occurring. And then after a while there's a decline, there's a death phase, or it's also known as senescence, where the microbes die off. And this is the growth pattern. This is what happens in a culture, a batch culture. All right, and that takes us to the end of chapter 9. Now let's take another little quick break time with Gizmo and Wicket and come back strong for chapter 11, the final chapter for exam 2. All right, welcome back from break time with Gizmo and Wicket. Let's hop right into chapter 11, the final chapter of this exam 2. Here we're learning about physical and chemical control of microbes. Remember, you need to know these terms: sterilization, disinfection, antisepsis, and decontamination or sanitation. You should know that sterilization is the gold standard. This is when everything that can cause disease is neutralized, like something that's been autoclaved or incinerated, right? Even endospores have been destroyed. Disinfection is one level below that. You have killed most microbial life, but not all. And this is on inanimate surfaces. Antisepsis, remember, this is on living surfaces, same as disinfection, but living surfaces. And then you have decontamination or sanitation. This is the lowest standard. This is just a mechanical removal of most microbes. This is the lowest stringency. All right, you should know that endospores are the most resistant and the hardest to get rid of, right? Endospores, and I suppose prions as well. These are the most resistant microbial types to treat. You should know that cidal agents kill. For instance, fungicides kill fungal spores and hyphae and yeasts. Virucides inactivate viruses. Sporicides kill endospores. Whereas static agents, they prevent from multiplying, but they do not kill. You should know the difference between critical medical devices, semi-critical medical devices, and non-critical medical devices. You need to know that the dynamics of microbial death. What you need to know is that microbes die in a logarithmic way. They do not all die instantaneously. They always die in a logarithmic way. You should know about surfactants. Remember, surfactants are part polar and part non-polar, so they can insert into the bacterial cell membrane and cause leakiness and cell death. You need to know what moist heat refers to. Moist heat could refer to hot water, boiling water, or steam. All of these are moist heat. You should know that hot water and boiling water does not reach above 100 degrees C. In order to go above 100 degrees C, you need steam and pressure, like what's found in an autoclave. And you should definitely know that boiling water is definitely not sufficient to sterilize. In order to sterilize with moist heat, you're going to need a autoclave with steam that reaches over 120 degrees C. You should know that dry heat includes heat from flame or electric heating coil, such as our incinerators in the lab. You should know that bacterial endospores exhibit the greatest resistance to disinfection methods. So oftentimes anything that can kill endospores is sufficient for sterilization, because anything else should also be killed by the time the endospores are neutralized. Remember this from lab. You need to know the difference between thermal death time and thermal death point when it comes to heat and killing of microorganisms. You should know that thermal death time is the shortest length of time required to kill all the microbes at a particular temperature. The difference between that and thermal death point is that this is the lowest temperature required to kill all microbes within 10 minutes. Okay, that's the big difference there. So please know the difference between thermal death time and thermal death point. And remember, boiling water, this is disinfection only. This is not sterilization. It will not destroy endospores. You should know that pasteurization is even less stringent than boiling water because less than boiling temperatures are inflicted. You should know that pasteurization is done to preserve the taste of beverages, right, and foods. It does not kill endospores. You should know that an autoclave allows you to reach 121 degrees C for 20 minutes at 15 psi. This is sufficient to sterilize different equipment. Again, dry heat, you should know that dry heat includes flames. Dry heat includes our incinerators in the lab. Dry heat can even include an oven. You see, like a hot air oven as well. These devices can result in sterilization, though it can take different amounts of time. You need to know that cold does not kill bacterium. Cold does not sterilize. Cold, if in a cold environment, this merely slows down the activities of most microbes. The microbes just freeze. But once you remove that substance from the cold, those microbes are reanimated. You should know what desiccation means. It's drying conditions. Next, we talked about radiation, and we explored some types of radiation in the lab, UV radiation. You should know that you can use gamma rays, X rays, and UV rays to try to sterilize. You should know there are two types of radiation: ionizing radiation, which causes breaks in the DNA, catastrophic mutation in DNA, and non-ionizing radiation, which causes those thymine-thymine bonds to form, those cytosine-cytosine bonds to form. So you're going to see ionizing radiation with things like X-rays and gamma rays, while non-ionizing radiation, this has more to do with UV rays. Okay, the next method we talked about was filtration. Filtration uses fine, ultra-fine filters to filter out microbes, though it's not perfect for sterilization because sometimes viruses can make it across the filter. All right, with that, we have reached the end of the exam review. I hope this helps. Make sure you study these handouts in detail and learn all of the material on the handouts. Refer to your book to fill in any gaps in your knowledge. Make sure you've done those dynamic study modules, the homework as well, and I think you'll be set. Study extra hard, and I wish you good luck on the test.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.